Okay, we'll give the students about a minute to log in. While we're waiting, are there any questions? Okay, are there any questions? I'm taking it, that's a no. All right, let's begin. Let's see, there's a few extra windows here. Shut that down. Let me remind you, we share my screen. Let me remind you that you do need to download Respondus Lockdown Browser and make sure it's running for your system before you begin a quiz. And I've got a link for actually getting it. But there's 2 practice quizzes you can take Wednesday of this week, meaning by tomorrow. Both of them will be good in the sense that it'll show you what my questions will be like and what my quizzes will be like. Practice quiz 00A will have we'll have essay questions. And that one you can take anytime on Wednesday, and you can take all day on Wednesday if you want to answer the questions. And they're essay questions. Practice quiz 00B is a multiple choice question. And you can take this quiz anytime between midnight, it's before or after, but let's say 12:01 on Wednesday and 11:59 p.m. on Wednesday. And it has to be done between midnight and 11:59 p.m. I can't remember, is midnight the next day or the last day? And it doesn't really matter. The point is that the quiz has to be taken at that time. And you can take it at any time, but it is a timed quiz. I don't remember how long you get, but once you start, it'll tell you. And you have 1 minute per question, and then you have a few extra minutes for the quiz, not for each question. So you have a little bit over 1 minute for each question. This one is multiple choice, does require Respondus Lockdown Browser. All right, any question about any of that?
Does it cost money for the browser?
No, it'll download it for free. You can download it. That's another thing I want to do. Where do I have that? There it is. You can get the link from the lockdown browser requirement. I think that's the one that has it. You should probably hit that link to get it formally, but it will eventually show here. Yep, there it is there. How to use it. There's a little movie there. And then the download instructions. And I assume that's how you download it. If not, it'll give you a link to download it. And like I said, getting help, really call the help center at Clark campus or email them and they'll walk you through changing the settings on your computer so the lockdown browser will work. Generally, most students, it will run on your system. In fact, I've never had a student who it never ran on their computer. It is true that it won't run on a phone and some notebooks it won't run on. But for a laptop or a computer, it should run on. The notebooks, I've got an option in there for it to run on all the options I can, but I don't think it runs on all notebooks. All right, I think that's it. I'll go back to here and start talking about the lesson. We're doing, oops, where is it? Lab 2 today. You need to read the textbook chapter through the end of the section on the compound light microscopy. Then you should read that before beginning lab 3, and you'll need to do that reading before lab 4 as well. I mean, read it before lab 3, and then it'll be good for lab 4. And we're doing aseptic transfers. And I'm supposed to talk about discussing and beginning the infectious disease project. So let me go back to the lab page. It's the infectious disease project. I think it's under lecture 2. Oh, it's under lab 2, sorry. The infectious disease research project. Click on this link and you will get the instructions, which I shouldn't have done it this way. I've got it on my computer, but we'll just go there now. That's funny. Anyways, we'll use this. It's a 100-point lab project, essentially a slideshow project, meaning what do you call that? A PowerPoint project. It used to be when we were in class, you give a presentation, but you just got the project now where you get the information and then write it up. Infectious diseases are an important part of our life. In this project, we'll look at this. You're going to pick a disease and focus on one specific infectious disease. It does have to be an infectious disease, and you're supposed to determine the underlying cause of the disease, which microbe causes it, how we treat it, what the social and economic and political factors are that affect our ability to deal with the disease. And you're going to become an expert on one particular disease. Okay, and this should help you for your future career in the health professions. You can read about this project format. Pick a disease, get it okayed with me because only one person in the class can have one specific disease. Although we can tailor it, meaning you can take one specific aspect, like somebody requested the flu and I told them that's fine. It has to be flu A, flu B, or flu C, meaning influenza type A, influenza type B, influenza type C. And I think influenza type A was the one that they chose. You must have a cover page which gives the title of the name of the disease, your name, the date, the course name. However, I don't want it to be a cover page. Put it as the first, on top of your first page of the text. Then you have the text. It will be worth 60 points. You will present your material of the text here. 15 points will be on grammar, spelling flow, readability. As much as possible, the text should be written in your own words. Whenever you're not using your own words, you should put it in direct quotes and say where you got it from. And some people tend to copy and paste information they find, largely from the internet, but it doesn't matter what your source is. And this material will be checked for plagiarism. And if the plagiarism is above 20%, I will look at it in fine detail to find out if you're copying and pasting. And if it's not in your own words, you'll get 0 for this project. Okay, so make sure it's in your own words and don't copy and paste. Anything that is not common knowledge should be either directly quoted, or, and then you state where you got it from, or it should be paraphrased in your own words. And even if it's, what am I saying? If it's not common knowledge, you should put in a reference for where you found that information. Meaning you should cite the information. And you don't have to cite all the information in a paragraph. If it's all coming from one source, you can put the citation, the in-text citation, at the end of that paragraph. However, if the paragraph has information which is not common knowledge, like you're giving, I don't know, different facts about a disease, each sentence should have an in-text citation if each sentence is getting a different source. Okay. In-text citations will be worth 10 points. So make sure you use the in-text citation correctly. What you should do is state the information and then in parentheses, state the author's name of the citation, comma, the year of the citation in the parentheses. Is that clear? And if it's more than one author, like 2 authors, you should have the author's first last name, meaning the author's last name, and second author's last name, comma, year of publication. If it's more than 2 authors, then you take the first author's last name, something like Smith, and then you say et al, comma, and then the year of publication, and then the end of the parentheses. Any questions about that? In-text citations and then the full text citations, which you have a citation page, which we'll just call the references. These are areas that students frequently lose points. Now in the full citation, you have more than just the author's last name, comma, and a year. You will have, let me go to here. You will have the author's full name. If it's only one author, it would be Smith. And then you give their initials. It could be something like JP for John Patrick, or maybe just J period. And then you give the year of publication. Then you give the source, and for a journal, it would be something like, oh geez, Journal of National Academy of Sciences, the volume number, the page number. And if you didn't give the year earlier, you can give the year then. But if you give the year already earlier, then you don't give it there. The full citation page has different styles, meaning depending on where the source is from. I gave it to you from a scientific journal, but it would be different for a book, and it would also be different for a web page. Any question about that? But give the full citation. Look up the citation style for the source. If it's from the web page, you just give the web page and obviously the author's name and the year. If it's from a book, you have to give the article name in the book. You have to give the name of the book, and you have to give the editor of the book, as well as the author of the book, who's given first, and the publisher of the book, and then the city of publication. And I may have left something out from the textbook, but that's about what it is for a textbook. Let's see. And then you use a citation style, and the plagiarism quiz taught you about the different citation styles. You can use any citation style you want to use, but make sure the full citation, as well as the index citation, all use the same citation style, meaning don't have APA for part of them, and then whatever MLA for the other part of them. And then the final report submitted in the folder in Canvas, which I think I've got that shown. It'd be this folder here. So click on that, and then you'll get this, and then you should have over on the right or down below a link that'll submit the paper. You must submit it in the formats that I require, meaning I got it. It'll accept a doc or a docx document, a rich text formatted document, or a PDF file. It must be one of those so that I can read it. If you submit it in another format, you will get 0 points for it, although you can submit it late, in which case you'll probably get 50 points for it, a total of 50 possible points. So make sure it's submitted in the correct format. That's why I've got it a specific file type, and that's to make sure that you make it in that file type, because some people have, I don't know what it is, it's called a Word document on their system, which I can't use because I don't have it on my system, my computer. I used to know the name of that, but I don't even remember the name of it. All right, any questions about the infectious disease project?
When and how do we submit our request for whatever disease that we want to cover?
You just email me, and then I'll check the list. As long as nobody else has it, you can do it. And as long as it's an infectious disease, I'll make sure it's an infectious disease before I okay it. So you just email me what your request is, and then I'll get back to you on that. And it's a first come, first serve. Like I think somebody's already requested tuberculosis and influenza A, but I think almost everything else is available. This tells you how it will be graded, and that is you have to have a section on the causative agent and then answer these questions for the agent. Obviously, only some of them apply to the causative agent. And then give a history, a section on the history, a section on the epidemiology, a section on the pathology, a section on the response and treatment, and a section on the socio, political, and economic considerations, as well as the formal full citation or references at the end. And I've got how it'll be graded down here. Come on. Sorry, that's not scrolling. No, it's got stuck. It's just not working. All right, so you get 1 point for the cover information. Remember, don't make a full cover page. 9 points on the causative agent information, 10 points on the history. Oh, darn. 10 points on the epidemiology, 10 points on the pathology, 10 points on the response and treatment, 10 points on the social, political, and economic issues, 15 points on the quality of your writing, 10 points on the in-text citations, 15 points on the literature citation page or the references, for a total of 100 points. Any question about any of that? All right. If not, let's go on to, and remember to use the email and the Canvas. I'll check that. Actually, Monday, Tuesday, Wednesday, and Thursday. And then Friday, Saturday, and Sunday, I'll check it every other day. If you check my Clark email, I will check it twice a week. And that's it. Okay. All right, I think that's it. So let's close down and go to my module 2. It's on aseptic transfers. Upon successfully completing this lab, you should be able to understand the purpose of using aseptic transfer techniques, determine if aseptic transfer was used from the data provided, meaning we'll give you information, and then you tell us whether aseptic transfer was used or not. Be able to define the following terms: what's ubiquitous, what is aseptic transfer, what's inoculation, and what is a contaminant. In our local environment, microorganisms are ubiquitous. This is problematic when working in a microbiology lab, because generally we are going to want to work with pure cultures. And that is a culture that contains one and only one species of microorganisms. Working with pure cultures and obtaining a pure culture was first developed by Louis Pasteur, who made aseptic transfers, or developed a way to make an aseptic transfer and then minimize the risk of culture contamination. But how he did aseptic transfers and got a pure culture was fairly difficult to do. And what he did was he did limiting dilution of a liquid solution, and he had to make up 10 replicas of one dilution, and he made different dilutions. And if only one replica out of 10 had growth in it, he concluded that he started that culture with only one microbe, and therefore it was probably pure. And if there was 2 or more replicas, he would say it wasn't a pure culture. Any question about that? Anyways, we'll learn how to make aseptic transfer. I think you'll learn how to do it in this lab, if not in the next lab. During inoculation, to get an aseptic culture, never lay the test tube cap or the test tube or the Petri dish lid on the workbench. If you do, you can get contamination. And that's because the lid is fairly sterile. And if you put it on the work surface, it's not sterile and it can pick up contaminants. Even if you put it upside down, things from the air can come into the lid. And then when you put the lid on top of your culture, it can fall out of the lid and into the culture. To minimize contamination, you want to tilt the tube or hold the Petri dish lid at an angle. I don't know if you can see this. And then have the lid partially over the plate. And then, I don't know if you can see what I'm doing because I can't see what I'm doing. And then work on this. Let me see if I can get it where I can see a little bit. No, when I'm sharing now, I don't see even any part of the screen. All right. Anyways, hopefully you can see this. And you want to hold the lid at an angle and then streak like on the
I
plate.
think you need to bring your hands up higher.
Yeah, okay. Like that? Can you see it
Yeah,
now?
that's better.
Okay. You're going to pretend you're not seeing the plate, but you're pretending I'm streaking in the plate. So do it like that, where the lid is over the plate and held at an angle so you can get in the plate. And this is showing you right here. And the same with the tube. You hold it at an angle and then inoculate it. And that reduces what can fall in because of, instead of the whole surface being open, it has to be, something has to fall directly here. If it falls here, it won't get inside the tube. Okay, and that's why we hold it at an angle. Obviously, it would be best if you were to hold the tube at 90-degree angle and then go in. And obviously with a liquid culture, you can't do that. Although with a solid culture, you could, if it was a test tube. Another thing you want to do is work efficiently and don't talk during a transfer because you have the culture open or the tube open. And then as soon as you're done, cap it. Okay, because while it is partially uncapped or fully uncapped and held at an angle, a contamination from the air can get to it. So you want to do it rapidly, and then when you're done, cap it so that it can't be contaminated. And if you work rapidly and then cap it, you will minimize contamination. And then obviously sterilize the inoculating loop before the needle, if you're using a needle, or forceps, if you're using a forceps, before you inoculate the tube, and you have to pick it up in a stock culture and then inoculate it. And then before you set this down, sterilize it before you set it down, okay? And then when you're all done, before leaving the lab, remember to disinfect the workstation, the workbench. And you should have done it beforehand to reduce the contamination in your environment. Okay, any questions about any of that? Where's my scroller? Oh, there it is. In order to inoculate, that's transfer cells to a new sterile media, you need to make sure that we sterilize everything that can potentially contaminate the media. Now, the media should be sterile before you use it, but you should have a Bacto incinerator for heating your loop, sterilize your loop. And there is something you can do to reduce the contamination of the tube. We'll talk about that in a minute. And then know the terms of this lab. What you can do to reduce contamination, pretend this is a tube and this is the Bacto incinerator. You take the cap off the tube. So we're going to say this is the tube. Take the cap off the tube, hold it in your other hand. So we'll say this is the cap. Hold it in your other hand. And then you take the lid and put it up against the Bacto incinerator. I'll try and show you where. Right there where it's hot. The hottest part would be right here. Put the tip of the tube right there. And that heats the air in the tube so it'll be flowing out. And that will reduce the contamination coming into the tube. You can read about that. All right, lab 2. You're not physically going to be performing these activities, but you will be watching videos on them. And we do expect you to know and understand the material in this lab module and in the videos. In procedure 1 and 2, you're going to be transferring a sterile liquid in procedure 1 using a glass serological pipette. In procedure 2, you're going to be transferring a sterile paper disc using forceps, and both of them are going to go into a test tube. So watch the video demonstrating proper aseptic transfer into a test tube. For procedure 1, transferring from a sterile, you're going to transfer sterile water into a test tube. And when you're measuring the water in a burette or in a pipette, it'll have indicators, markers on it, and the liquid will not be uniform across the surface. It'll form a meniscus. And how you read this meniscus is you read it not at the top of the meniscus, not midway through the meniscus or halfway through. You read it at the bottom of the meniscus. Okay? So this one would be slightly less than 1 mil, but we can round that to 1 mil because it's going to be 0.9, would it be something like 0.999, something like that, which would be rounded to 1 mil. Okay, any question about that? You should know that everything we use should be appropriately labeled. All right, so that's procedure 1. For procedure 2, you're also going to be using aseptic technique to transfer sterile paper disc into a nutrient broth. So read the procedure. And then let me go to the different results down here. I'm having a hard time finding it. So the liquid broth will look like this, and that is it's clear, transparent, or almost transparent. And that means there's no growth in it. For procedure 1, here, use this picture here, P1. You can see that it's turbid, because there's a clear one. And procedure 2, this is for procedure 2, the results. And procedure 2, these are the results, meaning you're not going to do it. We're providing the results to you and then giving you pictures. For procedure 3, I haven't talked about it yet. There's the results. In procedure 4, there's the results. Use this table for assaying your results. For procedure 1 and 2, if the nutrient broth is clear, the conclusion is no organisms are growing in the broth. Aseptic transfer was successful. If the nutrient broth is turbid, hazy or cloudy, organisms are growing in the broth. Aseptic transfer was unsuccessful. We'll come back to procedure 3 because I haven't talked about it. All right, let's talk about procedure 3. Procedure 3, you're going to be transferring Serratia marcescens into a test tube containing a broth. In procedure 4, you're going to be transferring Serratia marcescens to an auger slant. For procedures 3 and 4, watch the video demonstrating how to correctly transfer from a bacterial stock culture into sterile broth, that would be procedure 3, and into a sterile auger slant, that would be for procedure 4. So watch this video here. Some important notes about the video. You only need to watch until 4 minutes and 23 seconds into the video. After that point, you can stop the video. The video is showing you for E. coli, which is a white or off-white bacteria when growing. However, we're using Serratia marcescens, which is pigmented with a red or a pink culture. In the broth, it will appear pink, and on an auger slant, it will appear red. And so you can use the color to tell you whether Serratia marcescens is there or not. And then there's an error in the video, which there's a question about that in your worksheet, that happens between 3.5 and 3 minutes and 20 seconds in the video. Try and figure out what that arrow is. You can hear sizzling when he puts the loop into the E. coli, and you shouldn't hear sizzling. We'll just word it that way. Okay, so procedure 3, you're using aseptic technique to transfer Serratia marcescens to sterile broth. And in procedure 3, it'll look pink because we're going into a broth. Broth cultures are problematic because it's difficult to determine if contamination occurs because all the cells mix together in the broth. And if the contaminant is minor, it'll still appear pink from the Serratia marcescens. Is that clear? If the contaminant is major, it may not appear pink. Meaning if 90% of the growth is from contamination, it will be a different color than pink. All right, you should appropriately label the tube. Come on, losing my ability to scroll again, darn it. In procedure 4, you're going to be transferring aseptically Serratia marcescens on a loop onto a sterile auger slant, and there's a slant there. You're going to streak the sterile slant with an S-type movement up and down the slant, so the whole surface is essentially covered. I mean, obviously it's not covered there and there, but the majority of the surface is covered. Appropriately label it and then look for growth. Slants are better because if you have a contaminant, it will appear red on the streak, and then the contaminant will not be red. So it's easier to determine if you've got contamination on an auger slant than on a liquid broth. For procedures 3, if the nutrient broth is clear, no organisms are growing in the broth, aseptic transfer was unsuccessful, because remember, for procedure 3, we're deliberately putting in a culture. If nutrient broth is turbid, hazy, or cloudy, organisms are growing in the broth and aseptic transfer was successful if the organism has the correct color, meaning pink for a broth. For procedure 4, if the slant does not show confluent growth or a colony of growth in the streak, then no organisms are growing on the slant. Aseptic transfer was unsuccessful. If the slant does show confluent growth or colonies of growth on the streak, then organisms are growing on the slant and aseptic transfer was successful. And here are the results for procedure 3, the liquid broth, and then the slant for procedure 4. And then answer the questions. Don't answer it in the lab module. Answer it in the worksheet, which I have right here. Answer the questions in the worksheet. There we are. Question 0, describe the initial clarity of the nutrient broth prior to inoculating. State whether it's clear or turbid. Okay, question 1 starts here. And it's on procedure 1. P1 stands for procedure 1. Answer the 3 questions. And then the third one, you have 3 answers. So where there's a dot, there should be an answer. Then answer question 2. State whether it's clear or turbid. And based on these results, what conclusion can be drawn? Answer question 3 about procedure 3. State whether it's clear or turbid. Based on these results, what conclusion can be drawn? And then answer the question, how do you know whether you transfer the correct microbe? And you can use the color to answer that. Question 4 is the results of procedure 4. Is there bacterial growth on the slope? Answer yes or no. How do you know? Does it appear to be the bacterium you inoculated or contaminant? And then that's it for the worksheet. It looks like I got an extra page there. I should try and remove that. All right, if you notice, I have graded lab 00, and I'm starting to grade lab 01. If you did not get the perfect score on lab 00, you can resubmit it for full credit by this Saturday. And then sometime after Saturday, I will regrade the lab. Any questions about that? And hopefully during the lab, this lab here, I will be able to finish the grading of lab 1, but I've only got 1 or 2 students graded so far on lab 01. Any questions? If there's no questions, I'm going to stop the share, but I will stay here to answer any questions you have about the lab, and I will be here until 8:20 to answer questions. After 8:20, I will end this session.